Hello, brilliant minds! Ever wondered why nature loves sweet stuff like fruits, bread, and potatoes? Well, it's all because of carbohydrates, nature's very own energy packed fuel. And speaking of sweet stuff, here's a joke for you. Why did the carbohydrate go to school? Because it wanted to be a little sugar smart. Alright, let's get into the tasty details. Carbohydrates are basically polyhydroxyaldehydes or ketones, or their derivatives. That's a fancy way of saying they're molecules made up of multiple hydroxyl groups attached to either an aldehyde or a ketone group. Their general formula looks like C sub XH to O sub Y. Think of it as carbon plus water molecules arranged in a special ratio. Now, when we break carbohydrates down by hydrolysis, that means using water to split them, we get different products. This leads to their classification: monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides, like glucose, are the simplest sugars and can't be broken down any further. Oligosaccharides give you 2 to 10 of these monosaccharide building blocks when hydrolyzed, while polysaccharides like starch break down into many such units. But wait, there's more. Carbohydrates are also classified as aldoses or ketoses depending on their key functional group. Aldoses contain an aldehyde group, that's the CHO group, while ketoses have a keto group, which is a carbon double bonded to oxygen written as greater than C equals O. Imagine a flowchart laying these categories side by side with example structures. The visual makes it easier to digest, pun intended. So basically, carbohydrates range from single sugar molecules to complex chains, all playing vital roles in biology and nutrition. Next time you bite into a slice of bread, remember, you're munching a whole family of carbohydrates. Hi again! Let's zoom into glucose, the superstar sugar that practically powers life on Earth. Quick joke, why does glucose always feel so stable? Because it knows how to keep its rings together. Glucose is what we call an aldohexose, meaning it has 6 carbons and an aldehyde group. Its molecular formula is C6H12O6, which sounds complex but is basically 6 carbons, 12 hydrogens, and 6 oxygens. Glucose is special because it exists in 2 main forms, open chain and cyclic. In the open chain form, you can picture a straight chain of 6 carbons with an aldehyde group at one end and 5 hydroxyl groups attached along the chain. This is best shown with the Fischer projection, a flat depiction where groups hang off a vertical carbon backbone. But in water, glucose loves to fold up into a 6-membered ring called glucopyranose, named after the Payan ring. This ring form is created when the aldehyde group on carbon number 1 reacts with the hydroxyl group on carbon number 5 inside the same molecule, a reaction we call intramolecular hemiacetal formation. Interestingly, glucose doesn't stick to just one ring form, it flips between 2 isomers called alpha and beta D-glucose in aqueous solution, a process known as nutrition. Think of it as glucose changing facial expressions but staying the same sweet molecule. Picture the diagram showing both the straight-chain Fischer projection and the cyclic alpha and beta forms, with the ring and the hydroxyl groups clearly labeled. This dynamic structure explains glucose's versatility and why it's so important biologically, from energy metabolism to cellular communication. Hello, chemistry explorers! Time to dig into some cool experiments that prove what glucose looks like under the hood. Here's a funny start. Why did glucose break up with oxygen? Because it couldn't handle the reduction. Okay, bad joke, but reduction is exactly what we are talking about. When glucose reacts with hydrogen iodide, that's high, it reduces all its functional groups and yields an hexane. This product, a 6-carbon straight-chain hydrocarbon, confirms that glucose definitely has a 6-carbon backbone in its structure. Next, When glucose meets hydrogen cyanide, or HCN, it forms a cyanohydrin. This reaction directly shows the presence of an aldehyde group. It's like glucose waving a red flag. Then, when glucose is treated with acetic anhydride, it forms a pentacetate. Penta means 5, so this tells us glucose has 5 hydroxyl groups that can be acetylated, confirming the number of groups on the molecule. But here's the kicker. Glucose doesn't form a Schiff's base nor does it react with sodium bisulfite like typical aldehydes do. This indicates its aldehyde group is locked up in a cyclic form most of the time, not free as a straight chain. Imagine a neat table showing these reagents, high HCN, acetic anhydride, and their corresponding glucose reaction products. These tests give solid evidence for glucose's structure and behavior in the lab and in biological systems. Chemistry detective work at its finest. Greetings again. Let's crack the code on how to tell if a sugar is D or L and why that matters. Ever heard this one? Why do sugars never get lost? Because they always know their right and left. That's because in monosaccharides, the D or L configuration depends on the position of the hydroxyl group on the penultimate carbon, that means the second last carbon, in the Fischer projection. If the O on this carbon is on the right, it's a D sugar. If it's on the left, it's an L sugar. For example, D-glucose has the hydroxyl on the right at carbon 5 and its mirror image, L-glucose, has it on the left. Another cool fact, sugars called epimers differ from each other only at one chiral center. For instance, D-mannose and D-glucose look alike except their O hat carbon to differ. But here's a biological twist. Only D sugars are found naturally and active in humans. So sugar's handedness isn't just chemistry, it's biology's way of choosing the right fit. Welcome back, carbohydrate champs. Let's talk about how two sugar buddies link up to form disaccharides. Here's a light one. Why did the sugars get married? Because they wanted to bond for life. In chemistry, that bond is called a glycosidic linkage, a COC bond connecting two monosaccharides. Take sucrose. It's a sweet marriage between alpha glucose and beta fructose linked through a 1:2 glycosidic bond. That means carbon 1 of glucose connects to carbon 2 of fructose. Maltose is different. It's made of 2 alpha glucose units linked via a 1:4 glycosidic bond. This structure makes maltose a reducing sugar because one glucose unit still has a free hydroxyl group on carbon 1 available to react. Disaccharides don't stick around forever. They can be hydrolyzed back into their monosaccharide units using dilute acid or enzymes like sucrase. Hi all. Now let's travel into the world of long sugar chains, polysaccharides. Here's a fun fact. Polysaccharides are basically carbohydrates' version of a train with many carriages linked together. These carriages, monosaccharide units, are connected by glycosidic bonds forming long chains. Starch is a key player here. It's made up of amylose, which is a straight, unbranched chain with alpha-1, 4 bonds, and amylopectin, which is branched thanks to alpha-1, 6 bonds. Cellulose, on the other hand, is made of beta-D-glucose units connected through beta-1, 4 bonds. This means cellulose chains are straight and can form strong hydrogen bonds with each other, giving plants their sturdy structure. Glycogen looks like amylopectin but is even more branched. This highly branched structure makes it ideal for quick glucose release, and it serves as the storage carbohydrate in animals. Picture a diagram here showing line structures of starch, cellulose, and glycogen with different linkages clearly marked: alpha-1, 4, alpha-1, 6, and beta-1, 4. These structural differences explain why starch is energy storage in plants, cellulose makes up plant fibers, and glycogen stores energy in animals. So when you munch on a carrot or run a marathon, polysaccharides are hard at work. Alright, problem solvers, here's a real example to sweeten your chemistry skills. Imagine you have 1 mole of maltose, which we know consists of 2 alpha-D-glucose units linked together. The question is, how many monosaccharide units do you get after hydrolysis? The law or reaction you need here is maltose plus water, in the presence of an enzyme, yields to glucose molecules. In chemical formula terms, that's C12H22O11 added to H2O, going to enzyme action, produces 2 times C6H12O6. Looking at the masses, 342.3 grams of maltose plus 18 grams of water equals 2 times 180 grams of glucose. That balances perfectly, confirming that 1 mole of maltose breaks into 2 moles of glucose. Picture a diagram showing maltose reacting with water and enzyme, then the 2 glucose molecules formed. So in biology and industry, enzymes efficiently break sucrose, maltose, and other disaccharides down to sugars our cells can use for energy. This reaction is a key part of digestion and metabolism. Let's wrap up our carbohydrate journey with a quick recap. Carbohydrates have the general formula C sub X H2O sub Y, carbon combined with water in a special arrangement. They link via glycosidic bonds, which are carbon-oxygen-carbon bridges connecting monosaccharides like train cars. Glucose in water shows mutation, flipping between alpha and beta forms, changing its ring structure subtly but importantly. Hydrolysis reactions break down disaccharides into 2 monosaccharides using water, either by acid or enzymes, making sugars available for our bodies to use. Carbohydrates might seem sweet and simple, but they're the powerhouses behind energy in living organisms, from the food we eat to the fibers in plants. That's it for today's sweet exploration. Chemistry is not just formulas and reactions, it's the story of life itself. See you in the next session for more exciting science.